So today what we're going to be doing is taking a soil sample for microscopy. And what we're going to be doing is looking at the bulk soil instead of the rhizosphere, right? The rhizosphere would be about 4 to 6 millimeters away from the roots versus what's in the soil. So today we're just going to be looking at what's in the soil, not necessarily your soil microbes that are rhizospheric microbes.
Okay.
One thing that we're going to do is take a little temperature. That will dictate who's awake and who's not. And we're going to use a probe. Now, this probe is something that you can use, or if you don't have this, you can actually use an apple core, shovel. It doesn't matter as long as you get to that 4 inches, right?
Okay.
So we remove any of the top layer. Here we didn't actually really have any mulch here. And so we just press in, give it a nice little twist, and that's our core. A couple things that you can do while you're doing this is just observe what's going on. And you can see here that there's a clear layer of where, especially like in a no-till system where you're constantly adding more and more and more soil or organic matter, and right around here that stops. So we're talking about what, maybe 3 inches?
Yeah.
That you're seeing your horizon. However, where we want to take our sample is again at that 4 inches, and it's a range. So I'm going to go right here, and this is going to be our sample that we collect.
Okay.
Now, before I do that, we got to make sure that we actually properly label our container. In this case, a paper bag. I'm only using paper this time because we're taking the samples to get analyzed immediately.
Yeah.
Otherwise, I would not do that because these microbes can, in fact, eat carbon.
Okay.
Right? So we'll call this the tomato bed.
Yeah.
Our temperature is right at
70?
72? 71, right?
71?
72? What do you think? I have poor eyesight.
70... Maybe 71, yeah.
71, all right. And then, I don't think we need any other... tomato bed, Astarte, we know we're here. In a case where we were actually not analyzing it immediately, I would put more descriptive information. Okay. That's our core sample there. I'm going to put this in a little cooler with some ice in it so that they're resting.
Napping?
Taking a nap, yeah, exactly.
Cool.
And then we'll bring it and process it.
Okay.
Cool.
Very cool.
I want to start by introducing myself real quick. I am Ruben from NOFA, Mass, the Northeast Organic Farming Association. I'm pretty sure everybody knows what NOFA Mass is here. But for those that don't know, real quick, we are a member-driven nonprofit. We've been around for about, 50 years or so. This was actually all started with the Back to the Land movement people, trying to define what organic meant. So here we are. And I'm, first of all, thankful for actually AFT for allowing us to tag along into this workshop as well, and get the most eyes that we can on this as well. This portion of the workshop is going to be on soil microscopy. We're going to try to relate it directly to the no-till practices, perhaps with that lens of no-till. But before we get started on that, we will actually, real quick, this was actually, this workshop is supported by the Massachusetts Department of Agriculture and Resources, MDAR. They gave us a grant that essentially was to promote soil health through the microbial lens. And so we actually partnered with Microbial meter people. This is a tool that we'll discuss later too. To bring this tool to different farmers, show them how to use that, and also do microscopy samples of their soils as well. With the idea of implementation, right? MDAR is always about implementation so that farmers could implement practices. In this particular case, it's practices that will foment microbial communities, because we know that microbial communities are responsible for nutrient cycling, for disease suppression, and just ecologically speaking, we need these microbes for every ecological function as well, right? So to get us all started, and I excuse the small visual here, three things that we're going to be discussing today. Soil food web, rhizophagy cycle, to get us started as to what these things are first, right? So that we're all speaking the same language. The tools that we use to monitor for these communities. And then we're going to actually get into some of the microscopic samples that we collected here today. We collected 3 samples of this farm here, and we were lucky enough to actually have a conventional farm next door to collect samples from as well. So we'll take a look at those things and compare the two as well. And we'll get into details as we prepare the samples as to how we collected these, and how you may actually want to collect samples as well, and things to consider. I'm sure that most of you have actually seen this before. It's a very simplified version of what the soil food web is. And I'll go over it really quickly. It all starts with our beautiful sun shining light onto plants. These plants then actually photosynthesize. They use a lot of energy to photosynthesize, right? Takes a ton of energy for them to do that. And so that photosynthate that they actually are creating, up to 70% of that photosynthate is actually sent down to the roots of the plants, sent out as exudates. Those exudates are actually essentially liquid carbon, amino acids, organic acids, things that microbes may want to feed on. And so the reason why they do this is because plants understand the importance of the microbes. And so you would question them as to why would they actually send up to 70% of that very intensive thing to do that they're doing. Why are they doing that? So they understand the importance of it. So should we, right? Upon sending those root exudates to the soil, they are specifically partnering or fighting against 2 groups of microbes. Those are fungi and bacteria. Those can be then classified into different between species and functions as well. They could be decomposers, they can be mutualists, where they're actually associating with the plant, right? So we all know about mycorrhizal fungi, right? They associate with plants. Bacteria do the same, and we'll get into that in a second in the next slide. And so, these plants are actually attracting the bacteria, fungi. Upon attracting those, there's a thing called autoinducer. It's a chemical compound that all microbes release. This is their way of communicating. So these autoinducers are actually produced by these microbes to either communicate within themselves or other microbes in the soil. Those autoinducers are going to attract other microbes. Those would be the protozoa and nematodes. These are then the predators of the soil, right? These will actually consume bacteria, they'll consume fungi, thus providing that nutrient cycling, right? Because they're going to consume more than what they need. And so that excess nitrogen, everything has a carbon to nitrogen ratio, right? And so that excess nitrogen that they actually release now gets released into the soil in plant available form. That is nutrient cycling. And I have a video so you guys actually see that in action. So let's see what that looks like as well. And again, like every ecosystem in the world, you take your plains and you have your lions being the apex predator, or the orca in the sea. The soil also has these. And so if you keep on going up in trophic levels, you will actually find that as well. And that same nutrient cycling goes on and on and on. What are we looking for? We are essentially looking for a well-balanced soil food web that contains all of these things, plus more. Again, I said this is a simplified version of it, but we're looking for a balanced system where we're actually finding all of these in play so that we actually get all the benefits that I just mentioned, which is the nutricycling, the disease suppression, and all these ecological functions as well. Any questions about that real quick? Pretty simple, right? All right. I mentioned that I was going to talk about bacteria also partnering with plants, and this is really important here. This work, I have to actually go back to 2010, Queensland, Australia. They were doing a lot of work with plants. And what they found was that these little chloroplasts that you actually see here in the plant, which were once believed to be just chloroplasts, they found out they were actually microbes, specifically bacteria. And so fast forward to 2019, a group at Rutgers University, led by Dr. James White, discovered that that was just not an isolated thing where these microbes were inside of the plant. These microbes were actually forming what is called as rhizophagy cycle. Rhizo, root, phagy, eating, right? So now we actually have this information, which now we have to rewrite every textbook that you actually have read about on how the plant and microbe interaction. So what they found was you have these plants that are actually sending out the exudates that we talked about earlier, right? They're attracting these microbes. Specific plants attract their microbes. Specific plants may attract specific microbes depending on what they want and need. This is what they do. They could be considered sentient because they know what they need and want, right? And so the plant is dictating what they need. They call out for something. Let's say I want phosphorus. I'm going to get that solubilizing phosphorus microbe. I'm going to call it over here and say, hey, I think I need some of that phosphorus. Would you please care to share some of that phosphorus? So the microbe comes around to the root, and you can see the exudates over here. It comes to the root, the meristem root, and it enters the plant through the roots. Once inside the root, the microbe then gets hit with what's called NOx, or superoxide. That destroys the cell membrane of the microbe, leading it into this little chloroplast, into nothingness, right? So now the plant is extracting everything that microbe was eating. The plant now forms a gradient and kind of flushes it through the entire plant. Now, here's the miracle, if you will. The microbe then starts to actually push. That is how we get root elongation. We can't get roots to elongate. These hair roots do not happen in sterile environments. They only happen because microbes are doing it, specifically bacteria. They are literally pushing until they get expelled out into the soil. Now, before they get expelled, the plant goes, whoa, wait a second. You came in here with that thing that I just destroyed. You came in here with that flagella to help you, swim around in the soil. I'm going to give you all the genetic material that you need to rebuild yourself so that we can do this again. Hence, rhizophagy cycle. Very, very important to understand this because this is how plants developed to get their nutrition. They developed this way to fight against other things as well. So the things that we need to be considering as growers is what are the things that are going to shut down this rhizophagy cycle, right? Tilling can do that, right? We're talking about no-till here. Tillage can actually do that. Chemicals can do that. Fungicides obviously do that. Anything that's going to kill your microbes will do that. Not having the soil covered, mulching, is so important. And we can go on and on that, but I just wanted to present this as, okay, now this is our container. This is why we need to actually be protecting these microbes, right? All right, so real quick here, we're going to be talking about some of the tools of the trade. I have a huge bias towards this tool right here, and you'll see why soon. But there are other tools that are very accessible to the farmer as well. This is called the microbiometer. The microbiometer is a test that you can actually look at compost and soil. I think that you can actually also use it for compost teas and extracts as well. It's a colorimetric test. I have one right here. It has a nice little whisk. It has some reagents that you actually put into a little tube. Add enough water. Bounce. Put the reagent in there. Whisk it up. Once it's in solution, you actually put your soil in after it's been sifted. A little sifter. It's really a dish drain. You sift that up. You then collect your sample, a known amount. Add it in there. Sift it again. Tap, tap, tap. Wait another 20 minutes. And then you actually have a placard right here, which you actually add a card like this that has a color gradient, right? Then you add your sample onto that, wait 2 minutes, and record your numbers. It's basically it's a colorimetric test that's looking at the melanin of both bacteria and fungi. It will give you a bacterial and fungal ratio, along with the total counts of those microbes. It does not discriminate against what's alive or dead, right? And is only looking for microbes that are producing melanin. So just understand that. A lot of this work that's done, I know that Food Web School did a lot of work on succession, and what that ratio is telling you is where are you in the succession. So think about it this way. A soil that's been uncovered, weeds growing on it, definitely bacterial dominated soil, right? You then, which has a higher bacteria ratio than fungi. The opposite end of that spectrum is an old growth forest, where you would actually see a bunch of fungi and little bacteria. And so by looking at that, there's been a lot of research done saying, okay, well, if you have at least a 1-to-1, you're growing the kind of crops that you want to be actually consuming. That should be free of disease, higher nutrient density, things like that. So that's what the ratios tell you. So the numbers are presented in, so this example we have here is 412 micrograms of carbon per gram. So that's the unit that we're looking for, micrograms of carbon per gram of soil. And those numbers you can actually extrapolate to whether it's soil, whether it's compost, whether it's compost tea. They all have different values that you want to be having a minimum of microbial biomass. I would say the downside to this test is that it doesn't tell you what you have. It tells you how much you have. But let's say, for example, I do the test and I have a lot of fungi in there, but then when I look under the soil, it's just dominated by one specific fungus. That's probably not what I want to see. I want to see diversity, right? However, you could still use this as a tool to look at progression over time as well. Now, if you couple that with a microscope, now you're talking, now you're cooking, right? So that's what those numbers mean. All right, so I'm going to move on to some cool videos, actually, so that you actually can see what are the type of things that you may see under your soil. I'm going to start you guys with an easy one here. Is that moving? Yeah, it's moving. All right. So this, again, is at 400x total magnification. Any guesses of what we're seeing here? Nematode. All right. Do you know what type? Who called it out? All right. So real quick, pro tip on this. If you see this microbe actually bending its mouth like that, it's a bacterial feeder. Other nematodes can't do that because they actually either have a stylet or something in their mouth that actually helps them to eat other things, right? The exception to that would be a predatory one, which we'll see in a second. This is a bacterial feeder. You can see it actually has a conical shape mouth. It has a medium bulb and a terminal bulb, and that's how we determine that it's a bacterial feeder. So this will actually go and prefer bacteria. If you see this in your soil, you have the lion in the jungle, right? This is I wasn't sure if I was going to actually have access to a sample like this, so luckily we actually had a sample that we collected just today. Very similar area as to where the tomatoes were, where we were standing. But this is a video from my colleague Monique Bosch at Connecticut NOFA, where she actually took conventional soil under the microscope, just to see what that looks like. The conventional sample was just showing us, very mineral dense sample with no life in it, really. I mean, you saw the stuff that we saw earlier, right? That had life. That had no life in it. Maybe bacteria. But bacteria have no problem. I mean, bacteria can replicate in 20 minutes. By the time I actually have collected these samples, I'm pretty sure that I've had already a bloom in here, which is why I put them on ice to calm them down a little bit. But yeah, the bacteria is never an issue in any system, unless you're actually spraying the and killing them. I'll show you. I'm going to show you guys how I do this. This is not difficult at all. Like I said, my child actually knows where and how to find tardigrades. This is not a difficult thing. And, just a handful of times as you start seeing this, and actually, I'll give you an example. I was doing a workshop once, and the host was there, and I was explaining, all right, this is a hyphae of fungi, and this is this. Later on, we saw the same images, and he was able to actually point out, oh, that's a hyphae right there, right? Within 30 minutes of this turning a span. So this is not difficult stuff. We're only looking at a handful of microbes here anyways. And what I like to say about the microscopy is there's two tracks. One for the researcher that wants to know everything, to the point earlier, I have more questions. Somebody wants to know everything that's in there. And then there's, for the grower, all I need to know is shapes, colors, and diversity. Do I care what type of bacteria that is? Do I care what type of fungi that is? No. Are they different shapes, colors, sizes? We can all do that, as long as we have our eyes, right? So this is not hard work, and it's very fun and rewarding, I would say. So this sample right here, the way I prepare it is I'm using a displacement method where I actually take... Oh, by the way, these samples were all collected at 4 inches of depth. And they were taken from the bulk soil, not the rhizosphere soil. Rhizosphere would be right next to the roots. So this was actually bulk soil. There was no plant growing in there, right?
Distilled water?
This is actually, this right here is distilled water, distilled spring water. You can use distilled spring water or spring water. I often don't like to use this distilled water. The spring water is a little more comfortable for them. So I prefer to use the spring water. Do not use deionized water. That will rupture cell walls. So I added 2 mils of water, and I'm going to add enough soil to displace it to 1 mil, right? 1 mil equals 1 gram of soil. So that's all I'm doing right here. And so once I get to that 3 mil mark, I know that I have 1 gram of soil. I'll then bring this up to volume. We always start with a 1 to 5 dilution, right? And then we do this 30 times. Right. That's 30 times. One of the things that you can do is allow this to settle for a determined amount of time. I like the 10 seconds, whatever you decide to do, stay consistent, as that will determine who you're going to be seeing in that profile, right? So let's go ahead and actually just put this under the microscope. All right, so this first sample here is the sample where we first stopped at. This is the sample that had the charred parsley, I believe, and leaves.
Yeah, garden parsley.
This looks really dense. I'll put it on the microscope, but based on my experience, we may not be able to see through this. And we may need to actually go to a 10x dilution instead of a 5x dilution. When I collect my sample, I try to go right below the organic matter, pump it just slightly. The reason why I do that is because if I take the first draw, everything is going to be stuck to the sides of this pipette. So I want to make sure that I pump it so that I can get the microbes that I'm looking for. So 90-degree angle. Boom. That's pretty dense. I like to keep that in there just in case I have to go back to it. The other thing that we do is we take our cover slip, move it at a 45-degree angle back and forth a bit just to spread the sample. That way we're not looking at one slide that actually has all the material in that area. We drop it. May have to do a little bit of calibration here, but bear with me. We start with the 4x magnification. Yeah, that's really dense. See how you can barely see through that? We may need to dilute that further. Let's see.
Is that indicative of a lot of organic material?
It's indicative of aggregation. All right. I'm going to go ahead and scan this right here real quick. This is at a 100x total magnification. And again, remember the other slides we were looking at? You can actually see right through. Part of the reason why you want to dilute is so that you can actually see things. And it's very difficult to see through here. Let's go up to the 4 or 40, so 400. And it's almost impossible to see through here, right? So we're going to go ahead and take this sample and I'm going to go ahead and dilute that. What makes it dense?
Yes.
One
soil being dense.
Yeah,
One sample being dense and one sample not.
Yeah, there's multiple factors to that. Typically, a well-aggregated sample will show up under the microscope in the same way, right? It'll still have all the microbial glues, attaching everything together. You'll probably see more spaces in between because you're actually holding all those things. This was not holding everything together, right? And so that's what you're seeing there.
In terms of the soil health, it's not a positive or a negative.
No.
Now, as a geologist, I would see that and I would think you had a high silt or a high clay content that was causing that. But you're talking completely different stuff.
Yeah, so the texture does play a role in that. Whenever you see a lot of silt, you would see it under the microscope. And I'm sure that there's a lot of silt in here too. So silt does play a role in that. Clay, not so much. And we'll talk about clay in a little bit too, because clay actually looks just like bacteria. So yeah, we already mixed this up. We already broke up all the aggregates. I'm just going to give it a quick stir there. Let it settle for a second. And the thing about the sand, silt, and clay is that sand, silt, and clay can actually behave in different ways depending on the microbial glues, right? A sandy soil can actually act as if it was more clayey, but it's not, and it's just because of the microbial glues that are there. So, yeah. All right, let's let that settle. So we're going to hang out here at the 10X real quick, just to give a quick scan of this whole thing. But as we are here, remember how we were talking about the differences in color? This has a ton of folic acid, which is excellent. Folic acid can chelate minerals. So to the point earlier when somebody was asking you, are you amending your soil? Well, plants can mine for these soils, for these nutrients, but these folic acids will actually help deliver that to the plant.
Did you say folic acid?
Folic.
Like the dark souls?
Folic. No, the honey color stuff is a folic acid. Humic acid is this right here. And you have a lot of it. And folic acid is just a fraction of humus. And if you ask people what to define humus, you're going to get probably 10 different answers. But let's actually scan here on the 10X, right? When I like to get on the microscope, the first thing that I do is actually put it on the... Oh, what do we have here? No, nothing. Okay. Just scan, right? And who I'm scanning for are microarthropods. Oh, that's... is that something that we want to look at? Probably not. Microarthropods and also nematodes, because we would see those here. And look at that. Look at that fungal hyphae. That's a large fungal hyphae. Let's actually zoom in real quick. Fungal hyphae. So this is now at 400x total magnification. And you can now see that a little more clear. Again, we're going up and down into the column, right, just to make sure that we're capturing everything. But that is a nice, healthy fungal hyphae that you would typically see in a no-till system because we're actually not destroying the soil and breaking these little bits apart, right? All right, let's continue scanning real quick. And sorry if I actually make anybody dizzy. I saw some more fungal hyphae there, some there.
I don't really see a lot of movement, right?
Yeah, so we're not at the magnification to see a ton of movement. If we, oh, look at that. That's nice. We will get to 400x and hopefully see that. What I'm not seeing immediately are protozoa, but I have to say, all within context. What I'm observing this year is that the amount of rain is really damping diversity. And our stunted plants are not just because they're lacking sun, it's because we're actually selecting for very specific microbes in the soil because the oxygen is being diffused out of it. And so we're selecting for a lot of anaerobes. And if we don't have the right microbes for the other predators to eat, they're not going to be there. I mean, they will be there, but they will actually form a cyst. And that cyst, they'll break out of that cyst once the conditions are right. Next week we're getting over 100 degrees in temperature, so they're going to not like that either. Overall, this actually looks like a decent sample. I'm seeing a lot of these microaggregates. I'm seeing a lot of fungi.
Microaggregates, are they just the larger ones?
Yeah, exactly. A lot of fulvic acid, which, you know, people pay a lot of money for fulvic acid. And why pay for something if it's in your soil already? All right, so we're almost at the end of the slide here. And unfortunately, you know, nematodes, but that is not indicative of the soil or soil health. That's indicative of the conditions of the soil. Not having the predator there because we actually don't have things for them to eat. So let's see if we can actually find anything that's interesting around here. Maybe we can move on to the next sample, actually. But overall, I would say that this is actually. Let me get to the 400x2 actually, so you can see that. So just a couple features here. These are actually mineral particles. See those glass-like features? Those are mineral particles. These are those microaggregates that we were talking about earlier. Bacteria will flock to these. These are a nice habitat for them. And again, you want to see micro and macro aggregates, and this is a good indicator when you're looking for soil health to see these in there. I'm really curious to compare this to the conventional soil layer.
Just all the little jiggly balls, those are all bacteria, right? All the little small.
Only if they're perfectly round. If they're not perfectly round, those are clay particles. Also movement. Whenever you see movement, that could be Brownian motion. Brownian motion is a fancy word for vibration. This right here happening. It's just that it's happening at a much larger rate for them to, so you're perceiving that because we're looking at a very, very small area.
So seeing it moving, that doesn't tell you whether it's clay or bacteria.
No. Or if it's alive or not. I use a stain to determine that. There's two stains that I use, acrolein orange and methylene blue. That can, if you put this onto the slide, it'll permeate through the cell membrane, only to those that are actually alive, because it's staining the nucleic acid. And so if you do that, you can see what's alive and what's not.
So can you point out the bacteria?
Yeah, let's take a look at that. First you would have to focus, right? So we'll find a place to focus it. Let's do the middle here. And make sure that we have something in focus there, right? So that guy right there is a bacterium. And you have to observe it as I'm going in and out to make sure that it's actually round. This is my least favorite part, to count bacteria. I typically actually dilute it enough so that at least I have 30 per field of view. That way I'm not driving myself crazy. And I only use a quarter of the slide. And I put grids on as well. I have software I put grids on, so I can actually do that as well. But it's definitely my least favorite part, to actually count bacteria. Yeah, let me see if...
The density there is what?
So you see this right here? That's a clay particle. See how it's somewhat jagged? It's hard to tell. This is hard to tell. And this is actually acting up too. Anyway, so that's what that looks like. Question? Yeah, so that's a combination of organic matter, minerals, glues that are forming that, pieces of roux that will form those microaggregates. And you typically see a lot of bacteria clustered in there too. A lot of times they hide in there too from predators, and I've seen predators kind of go in there and look, searching around, going crazy for those microaggregates. All right, let's move on to the other field sample of the tomato plot. See if we can spot any similarities or differences, and then we can move on to the conventional plot. All right. All right, let's see. Oh, this one's interesting. Okay. Again, to our 10x or 100x. somewhat similar to the last one. We'll do a quick scan. We won't do the entire thing, just for the interest of time. But this is looking very similar to the last sample. Go a little deeper in here. I'm not seeing as many fungal hyphae as the last one. And real quick on that. The pH is more for bacteria. So bacteria are pH dictates the bacteria that will be present. Temperature, humidity, and those environmental conditions will select for fungi, which makes sense, right? Fungi like dark, cool places. If this was under a tarp getting beat by the sun, that fungi may say, not right now. Right now, I'm not liking this. Which is why we see a lot of flushes in the spring and the fall, right? Those are the conditions where we see more wet, humid conditions. And cool, right? That's the other thing too. They like it cool. They don't like it too hot. Only a few fungi like it hot. I would say the big difference that I'm seeing is that this looks more mineral, less aggregation than the previous one. And again, I'm not seeing all the fungal hyphae that we saw in the last one. So certainly less diversity. Well, amoeba, there's different types of amoeba, right? There's tested amoeba, there's arcella, and then there's naked amoeba. The one that I showed you guys earlier is a naked amoeba that doesn't have a shell. Oh, there's a fungal hyphae there. There you go. And amoeba are really interesting too because that shell, and actually this book right here has a really cool one there. So that's their test, that's their shell. And they usually make that shell out of their surrounding environment. And they're very particular too. I know that in composting, I'm going to switch over the sample. I know that in composting, they don't like to come out until like the temperatures are nice and cool, and so they actually form that test. But once that compost has matured, you would sometimes see the naked one because they're like, "Oh, okay, I can live in here. This is more hospitable for me." But they are finicky. The other set of protists, if you will, protozoa, are ciliates, amoeba, and flagellates. Ciliates will tell you whether or not your sample is going anaerobic because they like to live in low oxygen conditions, so you can use that as an indicator. So if you see those protists that are moving really, really fast, that have a lot of hairs around them, not a good indicator. Your soil's going anaerobic. The others, and if you see one or two, fine. But if you're seeing too many of them, that's a problem. All right, so I'm going to prepare the soil from next door. It was very interesting collecting that sample. The samples that we were collecting in this field, you can clearly see in the soil profile through the probe how there was clear organic matter. That's not it. That would have been bad. This one right here did not have any clear layer of organic matter. Same thing, we're going to break this apart. Without actually mixing this, you see how this actually just completely settled? Sand is what completely settles immediately. Silt after 5 minutes, clay 24 hours. So this is interesting, just an observation. But
it also appears that there's nothing in with the other one when you put the dirt in, there was still stuff in the soil.
not only that, look at the color.
Yeah.
We'll see, though. We'll see. We'll see what it looks like under the scope. But let's mix this first, too. And we can make and draw
I think they
comparisons.
did. This is next door.
This is next door. So I threw it in there undiluted. Or at the 5x, just to start with. And as expected, I think this is just more par for, to your point, with the type of soil that we're looking at. And not necessarily to do with the aggregation. Let's take a look at the 10x. Here it is. Yeah,
What's that?
that is an Arcella. So that's a type of amoeba. And you can actually tell by the vacuoles, food vacuoles. This looks like the organic matter plug right there.
What magnification is this?
This is 400x total magnification. Yeah. So that's interesting to see that guy there.
Would that be different from like a spore? The spore would that be?
A spore this size would be a spore of a mycorrhizae fungi, and it'll be way darker. And you couldn't see these features like the vacuoles and the organic matter plug. However, if you actually had an epifluorescence lamp, you can switch that over, shine the fluorescence on it, and see if it's shining. If it are fluoresces, it has that phosphate, which all fungi actually have. So you can actually definitively know that. But these features are not of spores.
What does that tell you?
It's a predator. It's a predator.
That's a positive.
It's a positive. It's a positive, yeah.
And
Like
this is,
maybe encouraged by the wet condition.
No, Arcellas and amoebas are actually microbes that like the oxygen. Here's the thing, and context is very, very important. This field appears to be that it was tilled recently before that crop went in. What happens in a tilled field, you mentioned this earlier, you get that explosion of oxygen. And so you see momentary brief benefits to the soil microbial life because you have introduced oxygen being the number one limiting factor that we all are facing. That quickly runs away when that oxygen gets depleted by that explosion, because now you have more bacteria respiring all of that, using all that oxygen. And what you see is, initially you say like, oh, everybody's happy, and then a sudden crash because they have used up all the oxygen in there, which tilling does, right? And you can see that in CO2 levels when you start to look at aerial maps around the globe and say, okay, well, when are they tilling soils around, the spring? Why are these numbers fluctuating so much higher? So that's what you're seeing. So the fact that I'm seeing that, you have to take all these things into context, really. You can't take a sample and say, well, you have this, you have that, so, great. What is the context behind that, right? Plus, it's just one organism. I would like to see 5 of those in order to say this is actually functioning.
Then we could
In a good sample, you'd say 5
5 of those, 5 of the other ones,
on the whole slide.
and the whole slide, exactly.
Just backing up that, something I noticed with no-till is that the soil stays colder longer because you don't have that introduction of oxygen and all of that respiration, like warming things up and getting activity going.
Yeah.
So like crop planning for that delay has been a learning curve, and then also like seeing plants kind of like go yellow and then green up, which I thought was kind of soil temperature related. So just that's been normal here, and it doesn't seem to change the overall health of the plants. It does delay some spring harvest, though. Yeah. And that's kind of with that mulch thing, I feel like wait for them to green out.
I'm just going to dilute this real quick. I know that we have to get going, but I do want to take a look at this a bit more diluted. That's the other thing too, right? We actually diluted all of them to 10x.
Mm-hmm.
This one we're just looking at 5x. So you may be actually artificially seeing more organisms too there because we diluted it less. Actually, not even more organisms. I didn't see any fungal hyphae in there, but we'll put it under the microscope again and see. All right, let's see. So this is now we can compare apples to apples because now they've all been diluted at 1 to 10, and we can now start drawing comparisons. The first thing that I want to draw attention to is how we're still, this is the same soil type, but we're still seeing that effect of this being so dense still. We can't see that anything clumping together. And so that's one observation. Let's just quickly scan through and see if we can actually find anything else here. Yeah, thought I saw something. Yeah, I'm not seeing anything now. Like this is
the Johnson seed compost.
what do we have here? It doesn't look like anything actually. At first I thought it was a springtail, but now, no.
What is that?
Oh, actually, that's interesting. Not sure what that is.
A blizzard?
Yeah, that's what it looks like, right? I mean, it does look like a nematode coming out of its egg,
What?
but it's not formed like a nematode. So I'm not sure what that is. It's not moving. Can't really tell any other distinguishing feature, really. Weird. All right, continue scanning. What's that? So we're basically looking for, larger. Same thing as for the first sample, we were looking for microarthropods. We're looking for nematodes, and at this magnification, we can also find fungal hyphae, which I have not seen any. That's a piece of organic matter. Yeah. So hyphae will actually present itself as uniform size throughout. It will also show very distinct cuts at the end. This is not fungal hyphae. It's just a piece of organic matter. Maybe a piece of root or something. Attached to an aggregate, though. But not what we're looking for. Ew, who are you? Ah. Oh, wait a second. Nah. I thought there was a tested amoeba. It's not. I'm not sure why it's green, but it could be algae. No, it can't be algae. We were able to see it. Maybe a piece of glass. Piece of Heineken. Yeah, this is barren in here. Yeah, I am not seeing anything that's exciting to dive into. We're not seeing the players of the sort of food web that we talked about earlier, right? And we can see some of this, some of the fulvic acid that we saw in the other samples too. However, not much in terms of microaggregation. There we go. What is this? That looks like a spore. There we go. Yeah.
So when we talk about carbon sequestration, where is that? Is it in here somewhere?
Any microbe is technically carbon, right?
It's in the microbes.
Exactly. Exactly.
But it's,
Oh wait. Oh wait. No, that's sorry. That was a mosquito.
I got full too.
But go ahead. Go ahead. This actually looks like an arcella.
Spore?
Yeah, it's not a spore. Sorry, it's an arcella.
Which is another type of amoeba.
Yeah.
We'll get pictures.
Yeah, so the carbon would be stored in the bodies of these microbes. And it's a cycle, right?
but it stays there. I mean, they die and then someone else takes them in.
They die, something takes it in, or they die and they become a part of the cycle, right? Of the carbon cycle. So weird empty spot right there. Nothing wants to touch it. That's strange. They're avoiding it too. Yeah. That is very strange.
That'd be like a spot where chemicals are or something, or
It could be.
okay.
It could be. But when I say chemical, it could be anything like a high
Yeah.
or high like a high salt chemical.
Okay.
So that could be, it could be a dead zone there because they're not going to touch that. Yeah, there's not much that I can talk about this sample here. It obviously doesn't have the soil food web like we were talking about earlier, and clearly lacking some microaggregation. I also want to point out the differences as well here, especially this sample in this one here, how it has had time to settle, but you can actually see some color differences as well. So, I think that concludes our visualization of the microbial community in the soil.